Well, good morning, good afternoon, good evening, whatever time zone you are in, you are most welcome here at the Organic Consumers Association. My name is Pat Thomas, and our topic today is the same topic for most of us every day: coronavirus, COVID-19, SARS-CoV-2, whatever you call it. It's the conversation that changes daily and these days almost hourly. And in addition to some of our more forward-looking concerns, like are we ever going to get our act together on testing, and will there ever be enough personal protection for key workers, and will the lockdown ever end, and in some parts of the country, will the water ever be turned back on again? There are now some more reflective concerns, looking again at the origins of the virus and asking, where did it come from? So over the next couple of days, we're going to be talking to scientists and to get their take on this. And with us today is Dr. Stuart Newman. Stuart is a professor of cell biology and anatomy at New York Medical College in Valhalla, New York. He's the editor-in-chief of the journal Biological Theory. He writes widely on the social and ethical aspects of biological research and technology, and he is the co-author of the recent book, Biotech Juggernaut. Welcome, Stuart.
Thank you.
So I think we've got a lot to talk about. I want to sort of just jump right in there, if I may. An increasing number of scientists, including yourself, are starting to speak up about the possibility that the novel coronavirus may actually have escaped from a lab. And that, even just saying that, opens up a number of different possibilities. Could you just take us through what those possibilities are about the origins of the virus and which ones you think are most probable?
Sure. Well, the most kind of suspicious looking thing is the fact that the virus was first identified in Wuhan, China, and that is the site of the only what's called a BSL-4, a biological safety lab for laboratory in China. And there's a BSL-3 lab, and research on bat viruses goes on in these laboratories. And the laboratories are not far from the wet market that is the most commonly attributed site of the origin of the virus. But I don't even think we have to even consider that market. So basically, viruses of this sort were researched in the lab, and not only by Chinese scientists, but by Chinese scientists in collaboration with scientists around the world, including in the United States. And the virus, after it was sequenced, was found to have some features that resembled earlier coronaviruses, things that were being worked on in the lab. But they also had some hallmarks of other kind of mixtures. And a chimera is something that's a mixture of several different types of viruses or even types of organisms. And this virus is chimeric. It has bits and pieces that look like it originated from viruses of not only bats, but possibly pangolins and other organisms that it might have passed through. So it's not like a pure virus, and no virus is. And so it has hallmarks of possibly having originated by manipulation, by being in different kinds of animals. The proximity to the lab is suspicious. So people have started to ask, could it have been partly a product of lab manipulation?
And are you saying that it could be a genetically engineered virus, or is this just part of sort of normal lab process to sort of mix up different viruses?
Well, these things can be partly genetically engineered, and they could have genetic engineering in their lineage and their history, and not be specifically genetically engineered for one purpose. So, for example, scientists work on different domains of the virus. There's something called the spike protein in the virus, and that's the protein that attaches it to cells and makes it so kind of pathogenic, makes it so dangerous. And the spike protein of this virus has a number of hallmarks that are very unusual. So it's very well suited to binding to something on human cells called the ACE2 receptor. So that's called the receptor binding domain. And the receptor binding domain is unusually avid for the human ACE2 receptor. And then it has something else, which is called a furin cleavage site. It's a weak point in the spike protein that makes it very flexible. So the way that you assay for that weak point is you use an enzyme called furin, and that will cut it. And almost no viruses in this category have that binding site, that enzyme cleavage site. It's something that where the enzyme cuts, and it's the weak point. And there are papers that show that genetic engineers working on viruses have introduced that by genetic engineering into other coronaviruses just as an experiment. So people do these things. It's not sinister, and it's not. And I don't think that this was for a germ warfare or anything like that. But scientists who are looking to make vaccines or looking to find out why these viruses are so dangerous make modifications, and they see in animal experiments if they become more dangerous or less dangerous. And there are papers in which scientists have introduced this cleavage site. It's a sequence of amino acids. They've introduced it by genetic engineering in viruses that don't normally have it. And it becomes much more dangerous when they do that. And this virus, very unusually, this coronavirus 2, SARS-2, has that site in it, and nobody knows how it got in there. So that is kind of another suspicious aspect. So it could have been that some experiment introduced it to a virus. The virus left the lab, maybe through an infected worker or a discarded animal, and then got into another animal and mixed up with the viruses in that animal, and then kind of came back to bite us in an indirect fashion. So it's not that you have to believe that some sinister scientist genetically engineered it to be such a nasty virus, but that it could have percolated through the natural environment after being genetically engineered.
It's interesting that you talk about how I'm getting a bit of feedback here. I don't know if you can hear that, but it's interesting that you talk about how the virus is constructed, because there was a recent paper in Nature Medicine that was talking about, this couldn't possibly be a lab constructed virus because of the way that it was constructed. They were talking about it not having the sort of expected viral backbone and things like that on it. And so they were using almost the same science to say, no, it wasn't from the lab, it was from nature. So how do we as lay people make sense of this sort of thing?
Right. So that paper, when I read that paper, it raised some red flags for me, because the people who wrote that paper made a very strong assertion about it being very unlikely to have originated in a lab. And what were the arguments they used? First, they said it has a very avid receptor binding domain, but it's not an ideal one. If scientists had designed it, they would have designed it differently. Well, that was kind of ridiculous to me, because there are papers going back 20 years that say that this domain is something that is very important to look at, and people should try a lot of different possibilities to see what it is that makes it bind. So the fact that this is not what other scientists have found to be ideal by computer programs doesn't mean that this wasn't manipulated. Then also, and they didn't refer to those papers that talked about modifying those domains. I was surprised. Then they point out that the furin binding domain, the furin cleavage domain is part of the spike protein. And they said this is not part of any other um, viruses like this. Well, that wasn't a good argument either, because they do refer to a paper where it was inserted by genetic engineers. Then the last thing was that they said that the experimental backbone, that if you started from scratch to try to modify a virus, you would take something off the shelf, which is called the standard backbone, and you'd modify it. And they said that this doesn't have the standard backbone. But in fact, people looking at these viruses take them from the wild, bring them into the lab, and kind of compare them to each other. And they don't always begin with the standard backbone. So they came up with this very unlikely scenario that somebody would try to make something really nasty and release it. And they said, well, this doesn't look like it happened that way. Well, I agree with that. But the fact that it didn't use the standard backbone is not at all a good argument. So and it's not only me, but a number of other people have found that that paper, although the research in that paper is very good, the conclusions that they draw from it are very speculative and uh, really raise red flags.
But imagine the frustration of average citizens who are trying to make sense of all of this, and every day there's a new conflicting opinion about where it came from or how it was constructed. Was it genetically engineered? Was it not? Um, it's really frustrating. I think if I can speak on behalf of the consumer, we just want to know that whether there's any certainty around the coronavirus, and it certainly doesn't seem like there is a lot of certainty.
There isn't, and I think that the honest conclusion is that there's a lot of uncertainty, but there are a number of red flags, and the red flags are that it was identified in Wuhan, which was a place where they were working on this kind of virus, that things like this have been genetically engineered in the literature. And then something I came across recently that I haven't seen written about is that there was a lot of controversy going back around 2010, 2012, about what's called gain of function experiments, where you take a virus and you try to add factors to its sequence uh, to possibly make it more dangerous, just maybe to use it to make vaccines or just to investigate why it's so pathogenic. And there are a lot of people lined up against gain of function experiments. They had scenarios like what we're going through now, and saying that if something like this got released by accident, it would be awful. So they banned gain of function experiments at the NIH. They didn't fund them, and that was in 2014. They put in that ban, and in December of 2018, they rescinded the ban. They started saying that as of 2000 and it was December 2017. They said as of 2018, they would begin to issue grants for gain of function experiments. Now here we are, a year and a half, after that, and we have something that looks like a gain of function. So I think that there was certainly research going on in the US and in collaboration with China that was doing gain of function experiments after 5 years of not doing them. So I find that very suspicious as well. And it was Anthony Fauci's institute that issued these grants and that rescinded the ban. So he kind of has an interest in minimizing the possibility that this is a gain of function experiment. But I think that one thing that we've found, and this is actually a theme of this book, Biotech Juggernaut, that I wrote with the historian Tina Stevens, we were looking at the prospect of human genetic engineering and all the kind of soft pedaling that was done to say that, if we did germline genetic engineering of human beings, it wouldn't be so bad. We could cure diseases and we wouldn't get any downside to it, and we found that very unconvincing. But scientists, particularly scientists that are backed by corporations, by patents, by business interests, and so on, have a strong stake in doing these things, trying them out, even though they might have adverse effects.
I think that's something that a lot of people don't realize is when they read in the newspapers about the latest research or the latest genetically modified whatever. That doesn't necessarily mean that it exists. It often means that the scientists are looking for funding, and so they're boosting it up all the time to say, "Look at how great this is! If we can just get this going, we can solve all of the world's problems." And so again, that creates confusion within the public mind and in the media about how useful something is, and why, if our scientists are saying this is safe, why is it suddenly not looking very safe?
Yeah, I think one thing that people should really understand is that there's this idea that's been hanging around for 50 or more years that we know how genes behave because we know how to sequence genes and we know how to study genes in a test tube. And we think, or some people think, that what genes do in an organism is each gene does a simple thing. But in fact, these are very complex systems. And if you put something simple into a complex system that wasn't there before, like genetically engineering a crop, for example, you can get all sorts of poisons being unleashed, that are just kind of weren't there before, but are induced by the new form. Let me give you an example, if we have some time for this. In about 2000, the year 2000, there was an experiment in Australia. They were trying to develop some kind of birth control gene therapy, some gene manipulation that would cause infertility. And they genetically engineered a mouse virus. And the mouse virus was innocuous. It was basically, it made the mice slightly sick, but then they put an extra protein in this virus, an extra gene that was already present in the mouse. They didn't put anything that they didn't know about. And then this virus killed every mouse that it encountered. It became absolutely lethal for all mice. So they stopped that experiment very quickly, but they had no idea that was going to happen. They just put a known gene into a known virus, and they got something completely unexpected.
Yeah. And I think that for me is if it turns out, we don't know. I mean, the evidence about the origins of the coronavirus is circumstantial all around, really. We're all sort of grasping at where it came from. But if it does turn out to be something that is genetically engineered, I mean, talking to scientists here, there doesn't seem to be any particular protocol for genetically engineering a virus. It feels sometimes like scientists that are working in that area are just kind of flying by the seat of their pants, which is not something you really want to do with something that's potentially lethal.
That's right. Right. Yeah. I think that's why a lot of people turned against this gain of function protocol, because even though there was a recognition that it could be useful, and you could develop vaccines with it, you could find certain principles by doing it. It was just too dangerous to try, and this is where we may have wound up having. I can't say that this is the way the virus came about, but it's as plausible to me as any other thing I've seen.
Sure. So I'm interested in sort of reading up before we spoke today on the state of play in terms of viral research. I mean, I think it's important. You made the point earlier, but I think we need to underscore it. It's not just China; it's global, and it is growing. And the number of laboratories that are doing this kind of work is also growing. And I guess my question is, as the number of laboratories engaged in this work on viruses grows, doesn't that also increase the risk of another, maybe even more catastrophic breach from a lab? I mean, where are we in terms of biosecurity?
Well, labs know what to do to contain these things, but since it's a living thing and it's invisible, and things can be breached, so it's not airtight. I mean, it's 99.9% of the time these containment protocols work. But if you have something that could be extremely dangerous, and there's a hundredth of a percent of the time when it might not work, and so I would say that it shouldn't just be at the point of potential release that it should be controlled, but at the point of design of the experiments. That's why, I would be an advocate for banning gain of function experiments. And since it's a little bit ambiguous because, as I said, we don't know what a gene is going to do when it's in a new context. So one person's gain of function is another person's just ordinary manipulation. So we just have to be super careful. We have to do things on a small scale, and we have to maybe increase containment when we're working with things like coronaviruses and other kinds of viruses that have caused problems in the past.
But that doesn't seem to have happened this time, does it? I mean, we're looking at the possibility that the Wuhan Center for Disease Control, which was the closest to the market, didn't necessarily have the security clearance that it should have had to be working with coronavirus. Isn't that right?
Well, they did have a BSL-4 facility, and there's a kind of an international standard for what that is, and the containment is very high.
I
People work.
understood it was BSL-2 in the one closest to the market.
Well, I
Is that
think the
right?
one closest to the market might have been, but the one that was doing the most exacting work was also in Wuhan, and that was a BSL-4. And the thing is that the market may not even be relevant. How close to it may not have passed through the market at all. I mean, it's when they try to identify, who it was that was the first patient, they don't find that there was a connection to the market. So I don't know about the market. I think in some ways, when I started looking into the market scenario, it sort of became like a xenophobic trope. It was like, this is kind of part of Chinese culture that we don't like or something. But I think it's more likely to have been in a lab and maybe inadvertently. And when you say released, it's not like somebody opened the door and the virus flew out. It's somebody touched something and walked out, with it, on a part of their skin or something, or there might have been an animal that somehow got discarded incorrectly or something like that. A leak could take many different forms, but I think that the fact that there was this high containment research going on in the same city is very suspicious to me.
Mm-hmm. So I guess I have a couple of questions left for you, and they're difficult questions, I think, in the sense of how do we weigh up the risks versus the benefits of this kind of research, whether we're talking about creating chimeras or genetically engineering viruses. The people who are involved in this research will say, but we're doing it so that we can prepare and that we can protect people. How does this research prepare us, and how does it protect people? Or is that just a line?
Well, I think that we have benefited from research, but bacteriological research and virus research. We've benefited in, I don't know. It's really, I don't think that there's an easy answer to it. I think that the research needs to go on. I think possibly, I think it has to go on in a more careful way. I think that the kinds of gain of function experiments that have been permitted, at least in the United States, for the last 3 years, I think they're going to revisit that. But we certainly have to keep looking into why viruses do what they do. I don't think that we should. There's no way that we can stay away from that kind of work. And there are things that kind of maybe lower level plagues, but affecting many, many people, things like Lyme disease, things like AIDS, that we don't have vaccines for. And it would be wonderful if we could get vaccines for these things. And it's except for doing this kind of work. In the case of Lyme disease, we're talking about a bacterium. That's not a virus. Maybe it's easier to control. It's more you can look at bacteria by an ordinary microscope. You don't need an electron microscope, or but I don't know. There's a kind of research that goes on for Lyme disease where isolates from patients are taken into laboratories. So, and then they're looked at and compared to see which ones are the most harmful and the most disease causing. Now, there's a case where you're not using a standard isolate, you're not using a standard backbone, you're using things from the wild. I mean, from patients. And there's a fair amount of virus research that goes on that way also, just isolates from animals, isolates from patients. So this idea about starting with one backbone and modifying it just doesn't happen typically.
Yeah. And I suppose related to that, I was struck by, it was an article this week in The Economist, and it was looking at the amount of research that has been published since the sort of identified beginning of the coronavirus pandemic. 7,000 papers have been published on the pandemic, and I think they said something like a fifth of them in the past week. We're looking to science. The mantra is follow the science, let the science lead us, let the science tell us what is right. How can scientists possibly keep up with that amount of information and come up with a coherent way forward for us?
Well, you're not going to read 7,000 papers in the space of a month. That's not the way it happens. But things get shaken out. What happens is that there's a kind of a crowd sourcing aspect to it, where somebody posts a preprint, and it seems to have something new, and then it kind of spreads virally itself, on Twitter and social media. But I mean, scientists. I mean, as a scientist, I look at Twitter to find out what other scientists are talking about, even before they publish papers on it. So, something came out in Science magazine just this week that said that this particular coronavirus has a lot of sites on its spike protein that have things called glycans. They're sugar groups attached to them, and there's very many of them. And these sugar groups both help it to bind to the human cells, but also protect the virus from being seen by the immune system. So it's kind of like a perfect storm. And so there's something about the spike protein of this particular virus that is similar to other ones, but is much worse. It's more protected and more avid. So, and that paper, not just because it was published in Science magazine, but because it seemed to have something new. It spread very quickly through the scientific community. So a lot of these other papers that are going to appear are going to be built on that. But I don't think there are going to be that many fundamental findings. But there's going to be a lot of variations and kind of mining of these new findings to kind of refine them. So there's a kind of screening process that scientists have that certainly doesn't involve consuming huge numbers of papers, but there's a kind of a crowd filtering process that takes place.
Okay. Science by social media. We're looking to you guys to solve this thing. Stuart, thank you so much for talking to us today. It's been absolutely fascinating, and we hope to keep in touch and maybe have you back.
Oh, great. Good speaking with you.
All right. Good speaking with you. Have a good rest of your day.
Thanks.
Bye.
Bye.